In our previous session, we talked about the immunoglobulins. But in this session, we will discuss the concept of alternative splicing. We will also discuss the germinal center reaction and the concept of hypermutation. Firstly, let's understand the concept of alternative splicing. As we know that B cell develops into plasma cell. These B cells are secreted as antibodies. When they are attached to B cell, they are called as immunoglobulins. And when they are released, they are called as antibodies. Immunoglobulins include immunoglobulin G, immunoglobulin A, immunoglobulin E, immunoglobulin M, and immunoglobulin D. However, heavy chain changes as B cell develops, so there are 5 different types of heavy chains, which encodes the isotypes. As we have discussed that classes of immunoglobulin are IgG, IgD, IgE, IgA, and IgM. These 5 are encoded by heavy chain genes, which are referred from a Greek letter: mu, delta, gamma, alpha, and epsilon. Initially, this cell express mu heavy chain. That's why B cell receptor is IgM. After this development, B cell undergo alternative splicing. This is the process by which cell express heavy chain for both mu and delta. That is for both IgM and IgD. So, both IgM and IgD are expressed on cell. But B cell is mature and naive. Naive means B cell has not been exposed to pathogen, and then these B cell pass from one lymph node to another. Let me explain the circulation of lymphatic fluid and blood through lymph node first. This is an artery. This is vein. It's the paracortex region. After the paracortex, there is cortex. Then this is the medulla. This is efferent lymphatic vessel. This is the afferent lymphatic vessel. This is primary lymphoid follicle. This is secondary lymphoid follicle. This is the germinal center. Lymphatic fluid enters the lymph node from afferent lymphatic vessels and leaves the lymph node from efferent lymphatic vessel. But blood enters lymph node through artery and leave the lymph node through vein. After entering lymph node, B cell enters the paracortical region first, and B cell then moves to cortical region. Now let's understand how different immunoglobulin are generated on B cell through germinal center reaction in germinal center that occurs in peripheral or secondary lymphoid organ such as lymph node. Unstimulated resting lymphatic follicles, such as those in fetal lymph nodes, consists of network of follicular dendritic cells in loose contact with small follicular B cell. That exhibits the surface expression of IgM and IgD. This is follicular dendritic cell. Once antigen contact takes place, secondary lymphoid follicles with prominent germinal center develops. The exponential growth of B cell takes place in germinal center of follicle in 3 to 4 days. After the contact with antigen, the cell develops into the large cell with large cytoplasm called as primary B blast, and small resting cells along with follicular mantle. A few days later, the blasts are concentrated in basal region of the follicle called as dark zone of the germinal center, where branching cytoplasmic process of the follicular dendritic cells form a fine loose network. These blasts are called as centroblast. These centroblasts have doubling time of 7 hours. They don't increase in number, since they quickly transform into small lobular nuclei called as centrocyte that migrate away from dark zone. These centrocyte then form the so-called light zone of the germinal center, where they come in close contact with dendritic cells, so a large number of centrocytes die due to apoptosis, especially near the boundary of dark zone and light zone, where numerous macrophages with apoptotic nuclei called as tangible bodies are located. The germinal center reaction lasts for about 3 weeks. Only few B blast cells can be found in the center of a burnt-out follicle after 2 to 3 months, called as secondary B blasts. So this is phase 1 of follicular reaction. This is phase 2 of follicular reaction. And this is phase 3 of follicular reaction. Now, let's revise the steps of germinal center reaction. Then we will discuss the antigen profile of B cells during germinal center reaction. In the germinal center reaction, we have discussed about the follicular B cell that converts to primary B cell, that forms centroblasts and then centrocyte. These centrocytes can undergo apoptosis or form circulating memory cell, or they can be converted to plasmablast. That can further either be the intestinal mucosa plasma cells or bone marrow plasma cell. Now, let's understand their profile. Follicular B cell has CD19, CD20, CD21, CD22, CD23, CD39, CD40, CD76, and IgM and IgD. Primary B blast has CD19, CD20, CD22, CD40, and IgE. Centroblast has CD19, CD20, CD22, CD38, CD40, and CD77, while centrocyte has CD10, CD19, CD20, CD22, CD37, CD38, CD40, IgM, IgA, IgG, and IgE. Centroblasts and centrocytes have high level of CD38 antigen expression. In contrast to follicular and extrafollicular B cells, they lost CD23 and CD39 antigen. Centroblasts also express a high density of CD77 because the transcription of immunoglobulin genes is temporarily halted and somatic hypermutation take place in centroblasts. The centroblasts are Ig negative. Centrocytes have renewed expression of immunoglobulin, which permits them to react with antigen presented by follicular dendritic cells. They may differentiate again into centroblast, but may also transform into memory B cells and plasmablasts, which then differentiate into plasma cells in bone marrow or in the mucosal lining of gastrointestinal tract. Now, after the generation of different immunoglobulins in germinal center, we will talk about the selection of high-affinity antibodies by hypermutation in germinal center. The aim of hypermutation is that to generate antibodies of different affinity. Centroblast achieves an extremely high mutation rate in immunoglobulin genes called as somatic hypermutation in order to generate antibodies of different affinity. Let's understand hypermutation in detail. This is the dark zone and this is the light zone. Centroblast converts to centrocytes. Centrocyte migrate to light zone of germinal center. Once there is only strong binding to antigen-presenting follicular dendritic cells, this binding can prevent them from undergoing apoptosis. As you can see, the centrocyte binded with follicular dendritic cell don't undergo the apoptosis. And the centrocytes that have not undergone the binding with follicular dendritic cell will apoptize. The centrocytes receive survival signal through CD40 from CD40 ligand-positive T cell in light zone. They then migrate to dark zone again and begin the new process of cell division as centroblasts. So, whole summary of this process is that mutated immunoglobulin with higher affinity bind antigen presenting follicular dendritic cells and survives. Now, the affinity of surface immunoglobulin for antigen can increase due to the point mutation. Substitution of amino acid. For example, as you can see this sequence in which there is thymine and adenine. But amino acids switch, as thymine and adenine has been replaced by guanine and cytosine. Amino acids switch can increase the affinity of immunoglobulin tenfolds. This mechanism can help to select B cell that produce high-affinity antigen-adapted antibodies. The demand for these antibodies determines whether B cell will able to survive and produce antibodies of desired affinity and specificity. This was all about today's session on alternative splicing and selection of high-affinity antibodies by hypermutation in germinal center. In our next session, we will talk about the important B cell antigens.